Dear students, good evening. Today I'd like to perform a brief idea regarding embryogenesis in plant tissue culture. You know what is embryo? Embryo is nothing but a plant in its initial stage of development. Embryo is nothing but a plant in its initial stage of development. This embryo may be the end product of sexual reproduction. The embryo is said to be zygotic embryo because gametic fusion takes place, zygote is formed, and the zygote is developed into the embryo. We are calling it as a zygotic embryo. In plant tissue culture, we are getting embryo from the somatic cells or the body cells. So the embryo is said to be the somatic embryo or somoclonal embryos or embryoids. This class, I'd like to give a brief idea regarding somatic embryogenesis. For embryogenesis, for the formation of embryo through plant tissue culture, especially somatic embryo formation, need an explant. The explant actually taken from the plant body, mother plant, plant, maybe from the shoot or from the root or from the node and internode of the plants. So the cell is to be grown in tissue culture is said to be explant or the inoculum. This inoculum is introduced into the culture medium where it undergoes rapid proliferation and form a massive undifferentiated and organized tissue called the callus. First, you are taking a cell from the mother plant. It is placed in a culture medium where it undergoes rapid proliferation to form the callus tissue. Then, for the induction of embryo, this callus is placed in another culture medium. If it is a dicot plant, the culture medium should possess 2,4-dichlorophenoxyacetic acid or 2,4,5-T, 2,4,5-trichlorophenoxyacetic acid to stimulate the growth of embryo. If it is a dicot plant cell, then you need different kinds of hormone like IA, indole 3 acetic acid, IBA, indole butyric acid, and cytokinin in the culture medium. So from the callus, embryogenesis takes place in mono as well as dicotyledonous plants. Initially, it resembles the globular stage embryo, followed by heart-shaped embryo, then torpedo stage. Finally, it resembles the real embryo of the higher plant. Here it is a dicotyledonous plant embryo. Embryo with radicle, the embryonic root, plumule, the embryonic shoot, and the cotyledons. This embryo, then placed in another culture medium, develops into plantlets. From the plantlets, we get new plants. It's a kind of propagation. Otherwise, this somatic embryo, because it is developed from the somatic cell, or the somoclonal embryo, or embryoid, is converted into the artificial synthetic seeds. The plants produced through the somatic embryogenesis are genetically similar to the plants. That means clones, genetically similar, genetically identical, genetically uniform plant populations are found through the somatic embryogenesis. And this embryogenesis does not involve sexual recombination. Sexual recombination takes place only in zygotic embryo because zygote is the end product of sexual reproduction. And the somatic embryogenesis is a powerful tool for increasing the pace of genetic improvement of commercial crop. So simple as the embryo developed from the somatic cells are called the somatic embryo, are embryoids, are somoclonal embryos. These are the plants are developed from the somatic embryos are genetically similar. That means they produce clones. Thanks a lot for hearing, students. Thank you very much. Thank you. Thank you. Thank you.